Okay. Continuing class on parasitology. So, parasitology is broadly divided into protozoans and helminths. Protozoans and helminths. Okay. Protozoans are further classified into amoebae. Amoebae is they are motile with pseudopodia. They are motile with pseudopodia. Example is entamoeba histollytica. It causes amoebic dysentery. It causes amoebic dysentery. Man get infected by the consumption of food and water contaminated with the quaternucleate cyst. Man get infected by consumption of food and water which is feco-oral route. Feco-oral route. Okay. Now demonstration is done by demonstration of this trophozoite cyst in the stool examination. Okay. Next is flagellated. So you have this Giardia lamblia. Giardia lamblia. Man get infected by the consumption of cyst by feco-oral route. Feco-oral route. Man get infected by consumption of food and water contaminated with the cyst. It gives rise to fat malabsorption. Fat malabsorption. And laboratory diagnosis again, stool examination. Of trophozoite and cyst. Stool examination. Next flagellate is genital flagellate. Trichomonas vaginalis. Trichomonas vaginalis. Please remember there is no cyst. Okay, it is a sexually transmitted parasitic disease. It is sexually transmitted parasitic disease. Man get infected during sexual intercourse. It gives rise to urethritis in male and vaginitis in female. It gives rise to urethritis in male, vaginitis in female. Okay. Here to lab diagnosis demonstration of trophozoite in vaginal discharge or urethral discharge. Demonstration of trophozoite. Please remember, there is no cyst. Entamoeba, Giardia has got cyst, whereas Trichomonas do not have cyst. Next is Plasmodium. Plasmodium vivax, Plasmodium ovale, Plasmodium malariae, Plasmodium falciparum, Plasmodium novelae. Novelae, Plasmodium novelae. Okay. The life cycle takes between man and mosquito. Man is an intermediate host, mosquito is a definitive host. Man is a intermediate host, and here a sexual cycle takes place. Here sexual life cycle. The man get infected by the bite of female Anopheles mosquito. Female Anopheles mosquito. Laboratory diagnosis is done by thick and thin smear, stain with Leishman stain. Thick and thin smear, and demonstration of erythrocytic stages, ring form, trophozoite, schizont, gametocyte. The quick way of demonstration of malarial parasite is QBC, quantitative buffer coat. So you take capillary coated with acridine orange, centrifuge it. In the buffer coat, you look for the efflorescence under ultraviolet microscope. Ultraviolet microscope. So in short, this is malaria. Okay, malaria. All this gives once in 3 days. Malaria gives once in 4 days. This is quarter. This is every day. No lacy every day. Fever every day. Okay. Next is Leishmania. Leishmania don't vary. So there is a promastigode stage and amastigode stage. The promastigode stage is present in dogs, rodents. Man get infected by the bite of sand fly. By the bite of the sand fly. By the bite of the sand fly. So they present in the reticuloendothelial system. They are present in the macrophages. The term mastigode forms is called as LD bodies. The mastigode form is called as LD body. Leishman-Donovan body. This you can demonstrate by doing Leishman stain. This you demonstrate by doing the Leishman stain. So Al-Azhar gives rise to black skin, and hepatosplenomegaly, anemia, hypergammaglobulinemia. So in short, you learnt about uh, entamoeba histollytica, then Giardia lamblia, Trichomonas vaginalis, then Plasmodium species, and Leishmania. So in short, you should know about sleeping sickness, trypanosoma, brucei. Man get infected by bite of tsetse fly. By the bite of tsetse fly, brucei, trypanosoma cruzi gives rise to Chagas disease. Bite of rudwit bug. Rudwit bug. Along with this, you are supposed to know one pig coli, Valentidium coli. Just like entamoeba histolytica, man get infected by eating food which contains cyst. Then there are free-living amoeba, that is acanthamoeba, and Naegleria fowleri. They are present in the ponds and swimming pool. Man get direct infection through the nasal inhalation when he is swimming in the swimming pool. It gives rise to primary meningoencephalitis. Primary meningoencephalitis. Primary meningoencephalitis. One more thing is Toxoplasma gondii. This oocyst is present in the cat species. Man get infected by eating food or water contaminated with cysts of Toxoplasma. Oocyst of Toxoplasma gondii. So this is about briefly about protozoal parasitic disease. Okay. Now moving on to the helminths are classified into cestodes, trematodes, and nematodes. Cestodes, trematodes, and nematodes. Cestodes are tapeworms. Trematodes are leaf-like worm. Nematodes are roundworms. Cestodes are the roundworms. Cestodes are the roundworms. So I give a best mnemonic for nematodes. You should remember there are two types again further classified into intestinal nematodes and extraintestinal nematodes. They are called as a visceral lymph node. Visceral nematodes other than the intestine. Visceral lymph nodes. Visceral lymph nodes. Intestinal nematodes and visceral nematodes. Remember yeast, mnemonic, yeast. Okay. E for entropius, vermicularis. Entropius vermicularis. A for ankylosoma duodenale and Necator americanus. Enterobius vermicularis, and Ankylostoma duodenale and Necator americanus. S for strongyloides, strongyloides stercoralis. S for strongyloides stercoralis. T for Trichuris trichura, Trichuris trichura, and Trichinella spiralis. And of course, A for Ascaris lumbricoides also. A for Ascaris lumbricoides also. So there are how many words? 1, 2, 3, 4, 5, 6, 7. There are 7. How many? 7 intestinal nematodes and 2 visceral lymph node. Visceral lymph node. One in lymphatics, that is Wuchereria, Bancrofti, and Dracunculus medinensis. Dracunculus medinensis. Again, Dracunculus medinensis is also called as guinea worm. Dracunculus medinensis is also called as guinea worm. Okay. Now, Enterobius vermicularis, it's also called as pinworm, seedworm, threadworm. Pinworm, seedworm, threadworm. Ankylostoma duodenale by consumption of fecal by embryonated egg. Embryonated egg. Roundworm. Enterobius Ascaris lumbricoides by consumption of food and water contaminated with the embryonated egg. Now, all three, the strongyloides, Necator americanus and strongyloides by penetration of skin. By filariform larva. Filariform larva. Filariform larva. Trichuris trichura, again egg. Okay. Trichinella spiralis is by consumption of uncooked pork. Uncooked pork. Uncooked pork. So, Enterobius vermicularis by egg, Ankylostoma duodenale by consumption of egg, duodenal americanus, strongyloides circularis, cercaria, penetration of when you walk barefooted on fecally contaminated soil. Trichuris trichura, again egg, and Trichinella spiralis is uncooked pork. Uncooked pork. Now, moving on to the lab diagnosis of this nematodes. So you have to do saline examination stool. In protozoa, you see cyst. For that, you have to do iodine preparation. Whereas in nematodes, you have to do the saline preparation, wherein you can differentiate bile stained and non-bile stained eggs. For example, this is you see 4 blastomeres, non-bile stained eggs. This is Ascaris, Ankylostoma, and Strongyloides circularis. Non-bile stained egg of Ankylostoma duodenale. Duodenal, Necator americanus, Strongyloides circularis. Then, so you can see the bile stained egg of Ascaris, bile stained Ascaris lumbricoides. Whereas non-bile stained without embryo, it is elliptical. Non-bile stained egg. This also non-bile stained egg of Ascaris lumbricoides. Non-bile stained egg of Ascaris lumbricoides. Now, Entropius vermicularis is plano-convex. And see embryo inside. Non-bile stained. Non-bile stained of Entropius vermicularis. Non-bile stained of Entropius vermicularis. So these are the eggs you have to see in for the diagnosis of nematodes. The treatment is wearing shoes or proper disposal of night soil and hand washing before eating food and eating hygienic food. And treatment for all that, there is a 400 milligrams of albendazole. Drug of choice for the treatment of all helminthic infection is 400 milligrams of albendazole. Okay. Cestodes, once again I'll repeat, there is a beef tapeworm, Tinea saginata, pork tapeworm, Tinea solium, and then dog tapeworm, dog tapeworm echinococcus, granulosus. There is a rat tapeworm, he corrected me last time of saying Google. That is Hemelepis nana. Right? Then there is a dog tapeworm. Hemelepis diminuta. There is a fish tapeworm. Fish tapeworm is Dracunculus medinensis. Sorry, Diphyllobothrium later. There is a Diphyllobothrium. So here man get infected by consumption of uncooked or partially cooked meat. Here man get infected by eating uncooked or partially cooked meat. And dog tapeworm, again eating food which is contaminated with the feces of the dog, which contains the egg. Again it is also fecal-oral. Here by eating fish, uncooked or partially cooked fish. Now one difference between Tinea solium and solium, man can eat egg in salad or partially cooked uncooked. It goes to brain. It gives rise to neurocysticercosis. Neurocysticercosis go to the brain and can give rise to epilepsy. Here it gives rise to macrocytic vitamin B12 cyanocobalamin intrinsic factor deficiency. It causes macrocytic hypochromic anemia. macrocytic hypochromic anemia here. Okay. Rest are all not very important. In trematodes, you just remember the names. It's not very important. There are two groups. One is cystosomes. There are three cystosomes: hematobium, mansoni, and japonicum. And fasciole hepatica, clonarchy sinensis. So, man get infected by the sarcaria. They have two intermediate hosts as a snail. Intermediate host as a snail. So drug of choice for all this thing is albendazole. Okay, albendazole, or can take ivermectin. Is not good. Enterobiasis, it's a pinworm, threadworm, seedworm. Okay, the enterobiasis is very simple life cycle. For example, it occurs in children. Okay, and it is present in the perianal region. It is ovoviviparous. Eggs hatch out into worm immediately. So in perianal regions, sometimes you can demonstrate the small worms. They are very small, like this, thread-like. They are small worms. In children, auto-infection is common, hand to mouth. They scratch the anal region, put it into the mouth. That is called auto-infection. Auto-infection, and it gives rise to perianal itch, or it is called as pruritus. It is called as pruritus. Hand to mouth, scratching the anal region, putting into the mouth, the eggs are put back, auto-infection. So as you said, it is small hair-like structure. It looks like a thread. Globally, 209 people infection, auto-infection, hand to mouth, overcrowding, impaired hygiene, personal nail biting, inadequate hand washing. So this is the egg, plano-convex egg, non-bile stained egg. The worm is present in the perianal region, goes to the mouth, cycle continues. Okay, it's very simple life cycle. You don't have to bother. Auto-infection, exogenous, by transferring egg to the mouth with the hands, scratched in the perianal region. Endogenous, retrograde migration of larva, hatched egg in the perianal region. Through ingestion, eggs, environment, surface of bed linen through contaminated fingers. Most of them are symptomatic, children, young adults, often symptomatic, males and older people. In the night, they migrate in the female and they give rise to vulvovaginitis in the perianal region. Eosinophilia, in all parasitic diseases, eosinophils are increased. So what you have to do is laboratory diagnosis. There is cellophane tape attached to the glass rod. You have to take cellophane tape, and it is called National Institute of Health (NIH) swab. It is kept as a spotter to you. NIH swab, National Institute of Health. That's the glass rod with cellophane tube. It comes. You have to take the anal swab, put it under the microscope, and see the worm. There are a few other points while considering the application. Number of systems, 4 to 6 consecutive days. Timing, sample should be taken. Chance the egg deposition is more. Late in the evening or patient has been sleeping for several hours, first thing in the morning. So early in the morning or immediately after. Female worms lay egg in the perineal region, in the rectum, hence eggs are detected in a stool examination. Adult worms, sometimes you can see the adult worm. I have seen a lot of adult worms. Children coming to the clinic, mother show the worms. So these are small thread worms, headworm worm, that is the egg, convex egg, non-bile state, convex egg, non-bile state. So this is a treatment. Drugs can be given: mebendazole, albendazole, parental pomade. The same treatment should be repeated after 2 weeks. Treatment of household members. Everybody should be in the house should be treated to eliminate asymptomatic or reservoir of perineal region, perineal infection. Prevention. Improving the personal hygiene. Improving the personal hygiene. Uh, Bed clothes, keeping nails short, clean, and frequent washing hands. Keep measures to contain transmission. Do hand washing, everybody. Steps of hand washing. Everybody repeat steps of one, one, one. Start doing one, one. First one, this one. Next two, three, four. Hey, that is not four. This is four. This is five. Six, six, six. One. That is two. One, two, three, four. This is five. Then six. Six. Hand washing. How many of you are carrying alcohol bus hand wash? Everybody should carry hand wash. Okay, question paper. Open the question paper. Let us see. Question paper. So, laboratory diagnosis of hydatid, they ask you hydatid cyst. Usually occurs in the liver. Okay, you have to do ultrasound or do aspiration. Then we do what is called as Cassini's test. In 1911, Cassini discovered this test. We take Cassini's antigen. Cassini's antigen. So it is injected. It's an intradermal test. And it is type 1. Hypersensitivity reaction. It is type 1 hypersensitivity antigen reaction. What is Cassini's antigen? It is Cassini's antigen is hydrated fluid. Hydrated fluid sterilized by seeds filter. Sterilized by seeds filter. It is Cassini's antigen is a hydrated fluid sterilized by seeds filter. Seeds filter. It is given intradermally into the forearm. You look for the redness, wheal and flare more than 5 centimeters. within half an hour, then person is positive for hydatid cyst. So everybody say Cassinis test. It's an intradermal test. Cassinis antigen 0.2 ml injected into the forearm. Within half an hour, you see the redness with wheal in the forearm, and you can the antigen is prepared from hydrated fluid, which is sterilized by seeds filter or any other membrane filters. Okay. Three dimorphic fungi. So you should remember the classification. You should not forget morphological classification of fungi. You should remember the mnemonic. Okay. Yeast like dimorphic, and molds. Yeast like dimorphic and molds. Example of yeast is Cryptococcus neoformans. Cryptococcus neoformans. Example of yeast is Cryptococcus neoformans. It is found in the pigeon's feces, and man gets infected by inhalation. It can give rise to pneumonia, just like tuberculosis, or it can go to the brain, can give rise to cryptococcal meningitis. Cryptococcal meningitis. Yeast like is Candida albicans. There are so many candidas, Tropicalis, Pseudotropicalis, Vishwanath, and all. Albicans is the most pathogenic one. Okay, and it can give rise to oral thrush in immunocompromised patient, and moniliasis in female genital tract. Moniliasis in the female genital. Dimorphic, you remember, they are yeast at 37 degrees, molds at 22 degrees. These are called dimorphic. Two morphology. Dimorphic means two morphology. Remember the mnemonic HBC. Histoplasma capsulatum. Histoplasma capsulatum. Blastomycosis. Blastomycosis. Coccidioidomycosis. Paracoccidioidomycosis. Paracoccidioidomycosis. Molds, you have Penicillium, Rhizopus, Mucar, Aspergillus. Penicillium, Rhizopus, Mucar, and Aspergillus. Then dermatophytes. Dermatophytes. Dermatophyte hold like this. There are three. Dermatophytes, remember as TEM. 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 Trichophyton. Trichophyton means it infects the hair, nail, and skin. hair, nail, skin, hair, nail and skin. Epidermophyton will not infect the hair. Microscorum will not infect the nail. Will not infect the nail. This they ask three dimorphic fungi, remember with the mnemonic HBC. Histoplasma capsulatum, blastomycosis, coccidioidomycosis, paracoccidioidomycosis. Remember the classification now? Yeast like dimorphic molds. Yeast, everybody say yeast molds. Yeast like dimorphic molds. Yeast like dimorphic. How do you cultivate viruses in the laboratory? Three methods. Sit down, don't stand up. Cultivation of virus in the laboratory. Simple. Animals, tissue culture, embryonated egg. Embryonated egg. Animals, tissue culture. Usually, suckling mice is used. There are tissue cultures, HeLa cell line, which is continuous, semi-continuous. Even human organs can be used to cultivate the virus. Embryonated egg, you've got a yolk sac, chorioallantoic membrane, allantoic membrane, and you can these are various routes of inoculation. Egg is kept as an applied exercise. Should draw the diagram of the egg and for an allantoic quality, amniotic membrane, amniotic cavity. So egg-derived vaccines are two: yellow fever and rabies vaccine. They are prepared in the egg. Yellow fever and uh, rabies vaccine. So three methods of viral cultivations are animals, tissue culture, and embryonated egg. Embryonated egg. Tissue culture. Three parasites that appear in the peripheral blood. Number one, Plasmodium species. Next is Leishmania-donovani. Third one is Toxoplasma. So these are the three parasites which appear in the fertilized egg of Ascaris, embryonated, roundworm with bile stain. It should show the embryo inside. Three viruses that cause common cold. You had COVID-19, then respiratory syncytial virus, influenza virus, H1N1. H1N1. Giardia lamblia, cyst and trophozoite, man get infected with the hemoflagellates. Hemoflagellates you have to write Leishmania, Donovani, then Trypanosoma brucei, Trypanosoma brucei. Trypanosoma brucei and hemoflagellates. Rhizopus. The difference between mucor and rhizopus is rhizopus has got roots. Mucor doesn't have roots. Mucor doesn't have roots. Draw the label diagram of HIV. You know everybody. I drawn and shown you. For you only, no? Yes or no? No or yes? I'll make it simple. Okay. It is a nice round draw. Inside it has a, and it has got two similar looking. Then it has got this is GP 120. This is GP glycoprotein 41. This is RNA. Similar two pieces of RNA. This is reverse transcriptase enzyme. This is P24. And one more layer there is there, P18. This is a diagram of HIV. Everybody should practice this. This is a diagram of HIV. Valentidium coli, it's a pig coli. It is ciliated. It has got cysts and trophozoite, and it is just like entomopathy. A man get infected by eating the lab diagnosis of HIV by ELISA. ELISA. What is the enzyme used in ELISA? Carbon test. Enzyme. What is the enzyme used? What is the enzyme used in ELISA? You have seen ELISA plate 96 well. The plate is coated with antigen. Okay. Antigen. Then you add serum, which contains antibody question mark. Then which is coated with enzyme. And add substrate, the color change. This is direct ELISA. Indirect ELISA, you take antigen, add serum, question mark, then add anti-human gamma globulin, which is coated with enzyme, add substrate, color change, that is positive. This is positive. Capture ELISA is, you take one antibody specific to that antigen, and put the antigen, you capture the antigen, one more specific antigen, and this is sandwich ELISA. Then add substrate, color change. This is sandwich, direct, indirect, sandwich. Okay, thank you.